Okay, so with this screencast, we're going to pick up again with retrograde transport, and what I'm going to do is walk you through the molecular details of how a COP1 vesicle is assembled and how that assembly is tied in with the KDEL receptor and the recruitment of KDEL sequence containing proteins. So what we're going to do is first introduce the main players. We've already talked about the KDEL receptor. But I'd like to introduce two proteins that, while their names are different, conceptually should be very similar to things we've talked about before. We're going to talk about ARF1, and what that is a small GTPase. And as we go through this, what you'll see is this is the equivalent for COP1 of the gene SAR1. And what we'll talk about next is SEC7. So what is SEC7? It's the GEF, the guanine exchange factor for ARF1. So what does that mean? This SEC7 is the equivalent of SEC12 in COP2 vesicle transport. So what we have is a small GTPase and a GEF, just if for COP1 vesicles, the same way we have for COP2 vesicles. So how does this work? So what I'm going to do is first just draw a giant cis Golgi. Obviously not to scale. I mean, if this were the size of the Golgi, your head would be the size of the moon or something equivalent to that. So what we're going to do is first so this is the KDEL receptor. And what I'm going to do is just draw a soluble KDEL protein KDEL interacting with the KDEL receptor. And when that happens, it starts to interact with SEC7. So the first set of interactions that we have to deal with are the following. One, KDEL receptor binds to the KDEL sequence. And that complex of the KDEL receptor recruits SEC7. So right at the beginning, we have the GEF for ARF1 interacting with the KDEL receptor. So we're already beginning to pull in all the things that we want to recycle, the KDEL receptor and KDEL sequence containing proteins into the region where we're going to build a vesicle. So what happens next is, let's just clear this out. So what's step 2? If we just look at it this way, so we're going to do a blow up of what's going on the surface of the cis Golgi. So this is the cytosolic side. This is the lumen, the cis Golgi. Right? So we have the KDEL receptor. we've got the KDEL sequence containing protein. I think in the notes I drew it slightly backwards. And then we have our sex 7. So that's where we left off. Now, I said that sex 7 is a GEF for ARF1. So what happens is ARF1 comes in. Let's see. So we have ARF1 in the GDP bound form, right? So this is essentially for all small GTPases, this is the off state. But when it interacts with sex 7, it comes in and goes out. What happens is GTP comes in and we load up ARF1 with GTP. So basically one nucleotide comes in, the other nucleotide goes out. So we kick out the GDP and replace it with a fresh GTP, just the same way you'd load a gun. You fire all the bullets, and by firing, I mean hydrolyzing GTP, and in order to reload, you need to get rid of the old nucleotide and put in a fresh nucleotide that can be hydrolyzed. So that's the next step. So what happens is that sex 7 exchanges the nucleotide on ARF1. So it loads it with GTP. Okay, so now we have sex 7 generating all this ARF1 GTP. What does that do? So let's just draw our membrane again. Just have the cytosolic side, the lumen side. And what I'll do is I'll just show a batch of these ARF1 GTP. So ARF1 GTP, when it's bound to the when it's bound to GTP, it's in the on form. And what does it mean for it to be on? While SAR1 flips out this hydrophobic peptide, ARF1 has a lipid modification that's kind of buried into the protein. And conceptually, it's the same thing. It pops out and anchors the ARF1 to the membrane. And so you get this cluster of ARF1 GTP right on the membrane. So you can see this is almost exactly the same as what we're seeing with COP2 vesicles, except now we're just calling it COP1. And I'm not going to draw the sex 7, but I'm just going to put in the KDEL receptor . And then we've got the KDEL sequence containing protein interacting with the KDEL receptor. So what's going on at this point is ARF1 in the GTP bound form associates with the membrane. So we've now created this landing pad for the next part of the molecular sandwich to start building the COP1 vesicle. So where do we go from there? The next step is pretty easy to remember because we're not going to go into every single molecular detail of it. I'm just going to draw the budding membrane. Right? So what we have is ARF1 all along the surface of the membrane. And that's in the GTP bound form. So I'm just going to note that with a T. Just to remind you that this color is ARF1. All right. And then in here, we have the KDEL receptor, and we've got our, and I'm not going to draw the sequence, but the KDEL sequence containing protein that's been recruited in there. So that's where we were last time, but what's bending the membrane? This is going to be really easy to remember. It's COP1. So the COP1 coat is made of several different proteins, all with Greek letters. So there's like COP1 alpha, COP1 beta. So there's a whole slew of these proteins that act together to form the coat. We're not going to go through all the molecular details. We're just going to call the whole thing the COP1 coat. And so what you see is that the ARF1 GTP recruits this COP1 coat. And that those proteins bend the membrane. Just the same way every other coat protein ends up deforming the membrane in order to form a vesicle. So this is the COP1 coat. So at this point, what we're seeing is the ARF1 GTP recruits the COP1 coat. The COP1 coat bends the membrane. And though I haven't talked about it, maybe I should extend this C terminus a little bit. Put in a third point. The KKXX sequence interacts with the COP1 coat. So the fact that the KDEL sequence has been, the KDEL receptor was interacting with SEX7 put it in the right place, but it really gets concentrated and anchored into the vesicle by the fact that that sequence is interacting with the COP1 coat. And so that's what allows this all to get concentrated into the nascent vesicle. All right. And then we finish budding. And again, we don't really go into the mechanism for how you finish budding off the vesicle. But we've got the KDEL receptor. We've got KDEL containing sequence proteins that have been concentrated in there. We've got ARF1 in the GTP bound form. I'm only going to draw a few T's. I have to take my word for it if they're all in the GTP bound form. And we've got the COP1 coat proteins, and we're just going to refer to them all as COP1. It's a single monolithic machine that has bent the membrane and eventually allowed it to form a vesicle. So I think at this point you can probably guess what's going to happen next. So the next step, and I'll just spell it out before I actually do it, we're going to get GTP hydrolysis, which is going to trigger COP1 disassembly. So when all these GTPs start getting hydrolyzed, what you end up with, the whole thing blows up, and you end up with an uncoated vesicle that's got the various cargoes in it. You know, KDEL receptor, KDEL containing sequences, that proteins that bound to the receptor, anything else that's a transmembrane protein that had KKXX would have been recruited into this thing. And then this goes on to eventually the uncoated vesicle, the uncoated COP1 vesicle, will dock and fuse with the ER. And this occurs via RABs, RAB effectors, and snares, the appropriate snares. So there's all sorts of different V snares and T snares, and there's ones that are specific for this process as well. But we're not going to go into that detail. It's more important to understand how the COP1 coat forms and how the process of forming allows it to concentrate the proteins and recycle them back to the ER. So before I end with this, I want to just talk about one last point, because it's kind of interesting and gets into how we think about what the identity of any compartment is. So I'm going to have the cis Golgi here. And so we've already discussed that this is where N-linked glycosylation occurs. And we've already discussed that this is where O-linked glycosylation occurs. All right. So what you would expect, based on everything I've told you, is that no protein in the ER should have O-linked, no resident protein in the ER should have O-linked glycosylation, because at least before this entire machinery that I told you, you would think that an ER resident protein wouldn't exit. But the fact that retention in ER is via recycling means there's a leak. So there's a small amount of ER resident proteins that will have N-linked glycosylation. But because these proteins can escape and then come back, there's a small percentage that will have N and O linked glycosylation. So somewhere between 5 to 10% of ER resident proteins will have O-linked glycosylation. And we now can understand that because what happened was that some of these, this is essentially the leak rate. This is the number, the percent that escapes and that get dragged back. And when they got to the wrong compartment, they were glycosylated, but then they were captured and brought back to the ER to face judgment for having trying to break out of the ER. So this is actually pretty good evidence for this idea of a recycling mechanism, on top of all the fact that we've purified and identified all the machinery that's involved in trafficking, both the forward, the anterograde, and the retrograde transport. But it's an interesting idea that you would have thought that these glycosylation events would be completely separate. But the fact that there's a recycling mechanism means that there's a small percentage, and we can estimate the leak rate based on the number of proteins that have both types of glycosylation, the percentage of ER resident proteins that have both types of glycosylation. And so with that, I'll end this screencast.